The first pathogen we'll talk about is coccus. Not the single pathogen. We'll talk about several pathogens, which are coccus. Coccus. We learn this meaning of this word. Coccus is it's a Greek word, originally written like this, coccus, which means berry or granule. It's like sphere-like structure. So coccus means the little balls, the berries. And in this chapter, we'll learn about three major coccus pathogens. Two of them are gram-positive, the Staphylococcus and Streptococcus. And we'll also learn about the gram-negative coccus, the Diplococci neisseria. Okay. And yeah, from today on, whenever we introduce any specific pathogen, we'll learn in a similar pattern. We'll learn several important information about the pathogen. First, we want to learn about their morphology and the method to culture them. And also we want to know their biological characteristics. And we want to study their pathogenesis. So we want to learn their virulence factors and also the disease it causes. So that's the clinical findings. And we want to learn the diagnostic laboratory test. So what type of method we can use to diagnose, to determine it's exactly this type of bacteria. And we want also learn the principles of controlling the infection. If you have some patient already infected by some pathogen, you want to know how to treat the patient, how to help them recover. And also you want to know how to prevent further infection. Okay, so this, for any specific pathogen, we want to learn this contents. Okay. The first one we want to introduce is Staphylococcus. In this name, Staphylo, it's also from a Greek word. Okay, meaning of this is a bunch of grapes. Yeah, you go to the grocery store, you see that bunch of grapes, how they look like. And coccus means granule or berry. So this is a bacteria which spherical structure, but the grouping of this bacteria is irregular cluster like this, like bunch of grapes. This is under light microscope. After gram staining, you can see the bacteria like this. And this is the scanning electron microscope image. You can see these bacteria grouped together to form irregular clusters. That's the Staphylococcus. And, okay, here are some typical characteristics. They are spherical cells. They group together like grapes, and they are gram-positive. So after gram stain, they show the blue-purple color. And they don't have flagella, so they are not motile, and they don't form spores. Here the spore means endospore, so they cannot form the dormant endospore. And sometimes they may produce capsules. The capsules, even in the lab, when we culture the bacteria while we shake the broth, they don't produce the capsule. But while they set up natural infection in our body, they probably will produce some capsule surrounding the cell. And that capsule is important virulence factor to prevent phagocytosis. Okay, when we culture Staphylococcus, here is a method and information related. This bacteria is aerobic or macroaerophilic. Do you still remember the meaning of aerobic? They like oxygen, right? Macroaerophilic means they prefer the low level of oxygen, okay? So they can survive at normal concentration of oxygen or low level of oxygen. And the colonies on the solid agar, solid medium, you can see that they are round and they are smooth. You can see that, smooth. And they raise from the surface. So it's like a dome like this, raised from the surface. This is different from E. coli. E. coli colonies are mucoid colony. It's like liquid. When the colony becomes bigger, they just flow around to the surrounding area. But Staphylococcus is not liquid-like. They form a dome like this. And also, glistening means that there is reflection of light like this, glistening. The best temperature for Staphylococcus is 37 degrees, which means that they have evolved with animals. They have already adapted to this body temperature. They are able to produce pigment. See here, the golden color of the colonies. That's because of the golden pigment they produced. This pigment is a lipid soluble pigment. It's not water soluble, so that the pigment only stick to the cell membrane, but they don't diffuse. The pigment does not diffuse into the agar because it's not water soluble. And also, it causes hemolysis, which means that it's able to lyse red blood cells. Here, this is a red blood agar. It contains the intact red blood cells. But surrounding the colonies here, this is a colony. But surrounding the colony, you can see a halo of clearance, which means that this part, the red blood cell, is completely lysed, so it does not show red color. So this is beta hemolysis. Just make sure that you don't misunderstand this picture. This like yellow-like color is not the pigment production. It's just the beta hemolysis, okay? The pigment will stick to the cell, to the colony. This is how we culture the bacteria. And the several growth characteristics. First is catalase positive. Catalase is an enzyme. This is a catalase structure. This enzyme is able to catalyze the breakdown of hydrogen peroxide. Hydrogen peroxide is a molecule, is a chemical which can kill bacteria. So in a hospital or in clinic, you can use 3% hydrogen peroxide solution to treat the wound, to kill the bacteria, to clean infection. But Staphylococcus is able to degrade hydrogen peroxide so that it can protect itself. It produces a catalase, and it can degrade this molecule to form water and oxygen. And because of the formation of oxygen, there will be a large amount of bubble. So this is an experiment we can do in the lab. Okay, on the glass slide. Okay, first you put the bacteria on the surface. You stick the bacteria on the glass slide. Then you drop the hydrogen peroxide solution onto the bacteria. If the bacteria is able to produce catalase, you will see a lot of bubble forming immediately. In several seconds, you can see the bubble. So that's the catalase positive bacteria. Or you can do the experiment this way. In the glass tube, you put the hydrogen peroxide solution into the glass tube. Then you use the cotton swab or a sterile stick to transfer some bacteria into the tube. And if it's catalyst positive, you will be able to see the bubble in several seconds. So this is a common biochemical experiment you can do. Staphylococcus is able to ferment carbohydrates, okay? But when it ferments its carbohydrates, sugars, it produces lactic acid, but it does not produce gas. How can you detect the production of lactic acid? Do you remember? If there is acid production, how can you detect the production of acid? Right, you can use a pH indicator, right? You add a pH indicator into the solution, the carbohydrate solution. When there is acid production, the color will change. Okay, so that's what we do. Okay, and it produces other antigens like the most important one is a protein antigen called Staphylococcus protein A, short for SPA. SPA is a protein produced by Staphylococcus aureus, but it can produce in two different ways. One is that the protein is on the bacterial surface. This is a bacterial cell, okay? So that the protein is a surface protein fixed to the surface. And we learned before that this protein is able to bind to the Fc region of IgG1, IgG2, or IgG4, the important IgG molecules. So that this is IgG Fc region. The protein binds to the Fc region so that this antibody cannot work anymore. And we know the IgG antibody can mediate the phagocytosis. The antibody binds to the bacteria and the Fc region binds to the Fc receptor, then it can initiate the phagocytosis. But here, this antibody is inactivated. It cannot mediate the phagocytosis anymore. The SPA protein can also be produced in a soluble state. The bacteria just secrete large amount of SPA protein. And this soluble SPA protein can also bind to the Fc region of IgG and can inactivate the antibody so that the IgG antibody cannot work. The phagocytosis will be inhibited. And another function of SPA protein is that it can also act as a super antigen to B cells. It binds to the receptor on B cell, and that can activate the B cell, which lead to the B cell apoptosis. It will kill the B cells. Once the B cells are killed, they are not able to produce antibodies. So that this SPA protein can inhibit or decrease phagocytosis. And Staphylococcus are also able to produce polysaccharide antigen. So that's sugar antigen, okay? Polysaccharide means sugar antigen. And they are also able to produce a capsid antigen. And even though, as I mentioned before, even though sometimes the capsid structure is not obvious, it might be very thin, but it can cover the cell and has some antigen molecule in the capsid structure. Okay, these are the important Staphylococcus species, and we'll focus on the first one, the aureus species. This is the we call it professional pathogen, which can cause infectious disease in humans. While the next two members, the epidermidis, dermitis, and also I cannot read this species name. These species belong to our normal flora, so they are on our skin all the time, and usually they don't cause problems to human health, but sometimes they might cause some opportunistic infection. And later we'll focus on the first species, the aureus. Okay. Here we'll first introduce their pathogenesis, their virulence factors produced by Staphylococcus aureus that include several important enzymes, toxins, and also their structural components. First, the enzymes. This bacteria can produce actually several important enzymes. The first one is coagulase. I think we mentioned this a little bit before. Coagulase is the enzyme which can convert fibrinogen into fibrin. I guess we learned this in biochemistry, right? Or physiology. So we know that fibrinogen is a soluble protein in our human blood serum. And it's soluble, but this coagulase is able to convert fibrinogen into fibrin. Fibrin is the insoluble fiber protein, right? So it will cause the blood plasma to clot, and the bacteria can hide themselves in the clot. Okay, this can protect the bacteria from phagocytosis. After the formation of plasma clot, the phagocytes, the immune cells, cannot get access to the bacteria, so that the bacteria can hide themselves in the blood clot. So this is the first enzyme, coagulase. The second enzyme is called hyaluronidase. Hyaluronidase. That's an enzyme which can hydrolyze the hyaluronic acid. Hyaluronic acid. I guess we mentioned that before. Hyaluronic acid is one of the most important components in connective tissue. Once the bacteria is able to degrade the connective tissue, they are able to spread, right? So this is important for their spread and invasion in our body. Hyaluronic acid. Another enzyme, okay, is the fibrinolysin. Okay, here the coagulase forms the fibrin to hide the bacteria in the blood clot, but they cannot hide in the clot forever, right? Because it's a fixed location. The nutrients in that specific location is limited. So if they want to spread, they want to invade other parts of the body, they need to escape from the blood clot. So they produce this fibrinolysin to degrade the fibrin clots so that the bacteria are able to spread to other parts. Next enzyme is nuclease. Okay, nuclease is the enzyme which can hydrolyze DNA molecules. And that can decrease the viscosity. I don't know whether I mentioned it or not. DNA molecules are the genetic material, right? They hide in our nuclei. They carry the genetic information. But in another way, the DNA molecule is a very big molecule. Once they are released from the cell into the extracellular space, DNA molecules actually they are very sticky. It's like glue structure, like the glue you use to stick the papers. So it's very sticky. DNA molecules can cause the extracellular environment with high viscosity. And that high viscosity can trap the bacteria in it so that the bacteria cannot spread. So we human evolved this ability to use extracellular DNA to lock the bacteria. But the bacteria evolved the ability to produce nuclease to degrade DNA into small pieces so that they can escape, they can spread. Very clever, right? Okay, the next enzyme is lipase. Okay, ACE means enzyme. Lipase means they are able to degrade lipids. So the lipase is an enzyme to hydrolyze lipid molecules. And we know that lipid molecules is on which part of our cell? Membrane, right? All the membrane, red blood cell, white blood cell. So they are able to hydrolyze lipid on the membrane, so they are able to lyse the cell and kill the cell. So if they are able to kill the white blood cell, they can protect them. Okay, so that's lipids. And also catalase. So the bacteria converts hydrogen peroxide to water and oxygen to make themselves safe. So these are the important enzymes produced by Staphylococcus aureus. And they are also produce several important toxins. Like, let me show you. They are able to produce hemolysin. Hemolysins. In this word, first part, hemo, what's the meaning of hemo? Blood, right? Lysin means to lyse the red blood, the blood cells. So they are able to lyse red blood cells. Okay, the red blood cells contain a lot of elements and nutrients. Once the red blood cell is lysed, the bacteria is able to get their nutrients. So that's hemolysin. The second toxin is the leukocidin. Those are the toxins which lyses neutrophils and macrophages. So this bacteria is weaponized with tools to fight against these immune cells, neutrophils and macrophages. Both of them are phagocytes, which are able to engulf the bacteria and kill them intracellularly. And also enterotoxin. The first part, enter, means, what's the meaning of enter? The gastrointestinal tract, right? So enter means the intestine or the gastrointestinal tract. And this is the toxin which can induce central nervous system effect with nausea and vomiting. And we mentioned before that this enterotoxin is heat stable, even though it's a protein molecule, but it's heat stable. And this toxin is resistant to the digestive enzymes in the gut, in the intestinal lumen, so they can survive in the digestive tract. Enterotoxin, but enterotoxin by Staphylococcus does not cause fever, so that's another property of this. The next toxin is called exfoliative toxin. This is a toxin which can separate the epidermis from the dermis layer of our skin. Okay? Especially for the newborn baby, their skin is so weak, and if the bacteria cause infection in the skin, it produces this toxin. It can peel off the epidermis layer from the dermis layer, and that can cause a syndrome called Staphylococcus scalded skin syndrome. Later, I will show you the picture of this syndrome. Okay, the last toxin we want to mention is a toxic shock syndrome toxin, which can induce fever, vomiting, shock, and also systemic organ failure. Here are several important toxins produced by the Staphylococcus. Okay, the structural components, we mentioned the SPA first, but they are also produce other structural components like the capsule, which can inhibit phagocytosis by neutrophil and also facilitates the adherence. And it's gram-positive bacteria. It has thick peptide glycan layers, and that have endotoxin-like activity. It's able to activate the complement system. And also, it's on the gram-positive cell wall. It contains the teichoic acid. It's a polymer, okay? And the teichoic acid binds to the fibronectin and mediate attachment. This is one function of teichoic acid. Recently, in our lab, we published a paper on Staphylococcus teichoic acid. We found that this molecule also contributes to antibiotic resistance. And once the modification of teichoic acid change, the bacteria will become more hydrophobic, so that the water-soluble antibiotic cannot kill them anymore. So they will become more resistant to water-soluble antibiotic. But meanwhile, they will become more sensitive to the hydrophobic antibiotic. So that's a paper we published recently. It was online a week ago. Yeah, about that, a week ago. So if you want to find the paper, you can help yourself to find the paper. And also, they are able to produce protein A, and which binds to the Fc region of IgG antibody and inhibits phagocytosis. We introduced in detail just now. Clinical findings. So that's a disease caused by Staphylococcus infection. Staphylococcus infection can cause two types of disease. One is a pathogenic infection. That's due to the bacterial infection itself. Another one is a toxigenic infection. So this is the problem due to the production of toxin, not the bacteria itself. Okay, let me show you the common pathogenic infection. It can be cutaneous infection on the skin. It can also be systemic infection or deep in our body. First, let's see localized cutaneous infection. Because of the bacteria invade into our skin through either wounds, follicles, or glands, it can form infection on our skin. For example, like this one. That's due to the Staphylococcus infection, or the follicle infection, like this on the skull, or this one. This is a gland, okay? There's a gland underneath the eye, and the bacteria get into it and set up local infection. It has a special name. It's called stye, S-T-Y-E, stye. Probably you might have some problem like this when you were very little. So So because it's common in little kids. Has anyone suffered from this before? No? You? Yeah, it's not a serious problem. You can apply the antibiotic gel into the eye. It can kill the bacteria, and usually the kids can recover in a short time. But if the problem becomes serious, the doctor might need to drainage the part, need to open this part, clean the bacteria out of it, and apply antibiotics. This is the pathogenic infection on the skin, but pathogenic infection can also happen in internal organs, like in the heart, okay, or in the lung, or even in the brain. Okay, that's deeper infection in our body, such as in the heart, the bacteria might form biofilm. That's a serious problem. And it can also cause the systemic infection to cause the septicemia, pyoemia, that's systemic infection. Okay, the second type of infection is toxigenic infection, or we call it toxinosis. So this the disease because of the production of toxin, such as the food poisoning, the enterotoxin we mentioned just now. This toxin is characterized by a short incubation period. So after 1 to 8 hours, and it can cause violent nausea, vomiting, and diarrhea, but it does not cause fever. So that's the enterotoxin. And also toxic shock syndrome. I'll just leave it for you to read this part. And it can cause the Staphylococcus scalded skin syndrome, short for 4S. What is the meaning of scalded? Does anyone know? What's the meaning of scalded? You know that? If you work in the kitchen and accidentally hot water get onto your skin, right? So that's scalded. So this is characterized by the separation of epidermis layer from the dermis layer. Let me show you a picture of this syndrome. This is a syndrome due to this toxin. The newborn baby got infected by Staphylococcus, and the toxin can peel off the skin, the epidermis layer from the skin, okay, leaving the scalded structure, looking moist areas like this. We are talking about the first gram-positive cocci, Staphylococcus. And last time we introduced the virulence factors and the clinical findings they cause the different type of diseases. And here we'll continue with the laboratory test. So these tests are kind of the standard test we follow in the lab. After we receive the specimen, we run several tests to collect the information to help the doctor to decide whether it's a staphylococcus or not. First, the specimen collection. And last time we mentioned that staphylococcus can cause different type of infections, both the cutaneous infection and also deep infection in the deeper organs, and even systemic infection. So there's a lot of material, a lot of specimens you can use, but based on your patient's real situation, such as the surface swab, use a sterile swab to collect the specimen, or you can aspirate from the abscess, okay, the bloodstream, tracheal aspirate, or the spinal fluid. All these are possible specimens, but you don't have to collect all this from any specific patient. You want to decide based on the actual situation. Once you receive the specimen in the lab, you want to have a quick check. So that's what we learned last time. Stain the specimen, check it with a microscope, and you will see the typical gram-positive cocci in irregular clusters. So that will help you to get some very important information that this is Staphylococcus. To culture the bacteria, we rely on blood agar, especially in the hospital. They use blood agar a lot. But in our research laboratory, we don't use blood agar to culture the bacteria. We use agar called TSA or TSB agar. It takes about 18 hours, so that's usually it's overnight. We culture today, and you will see the results tomorrow. 18 hours at 37 degrees, so that's the body temperature. And okay, you can see the colony on blood agar. It's smooth colony, and also it produces the golden pigment, which is liquid soluble. And you can also see the beta hemolysis, the complete clearance of red blood cells surrounding the growth. And as aureus can ferment the molecule called mannitol. This is my mannitol molecule. So only this species is able to ferment mannitol, but other staphylococci cannot ferment mannitol. This is a very useful experiment to differentiate Staphylococcus aureus from other species. Okay, and the doctors usually run the coagulase test because we know that if the bacteria is able to produce coagulase, they usually cause serious problem in patients. So that's a coagulase positive staphylococci. So we do this experiment in tubes like this. So you mix in the tube of rabbit or human plasma with proper volume of broth culture. You culture the bacteria in broth. You can use the broth culture with a supernatant, mix it in the tube, and then wait for several hours. Then you check the texture of the material in the tube. If it's solidified like this, it's like agar. It's not liquid anymore. We say that the broth contains the coagulase, so it's coagulase positive. So this is coagulase positive. If in the tube is still liquid, flows, then we know that this is coagulase negative. The coagulase positive staphylococci are considered more pathogenic for human. So which means that they can cause more severe disease. But actually in the hospital, the doctors are now paying more and more attention to the coagulase negative bacteria, coagulase negative staphylococci, especially the opportunistic infection from the normal flora. Even though they don't cause severe disease, but they might cause a chronic disease, like we mentioned before, the prosthetic joint infection. A lot of times it's due to the coagulase negative staphylococci infection. So both of them can be pathogenic. This is a coagulase test. To treat the staphylococcus infection, we should have, first we need clean the infection site. as soon as possible, as timely cleanliness, and aseptic management of the lesions. For example, if you have some swelling infection on the skin, you want to clean it, treat it with aseptic material such as antibiotic or hydrogen peroxide to clean the infection site. And then for the abscesses and other closed separating lesions, if the lesion is not open, it's closed, the doctor probably need to run the surgical drainage. What's the meaning of surgical drainage? They need to first cut it open, right? Cut it open, then use probably saline solution to rinse it so that to wash the bacteria out of it. So that's surgical drainage. Like the sty I showed you before, the gland infection underneath the eye. So that's a closed infection. If antibiotic gel is not working, they need to drainage the infection to help the patient. And we should use the appropriate antibiotics. And we can perform the antimicrobial susceptibility test before choosing the drug. So you want to use the proper antibiotic so that it's more efficient, and also we can save the antibiotic which can cause selection pressure for other microorganisms. So that's very important for uh, medicine management. And if your patient is suffering from systemic infection, that requires intensive therapy. So probably longer time and more careful treatment or therapy. The staphylococcus sometimes can cause serious problems.